Okay. Now, I start with a little premise. A little premise is like this: life and, in general, living systems are multiverse, which means that the apparently contradictory features are present at the same time in the same object. This is a very important thing to say, for you have not to, if you don't say this, if you don't think about this fact, well, you can choose one of the two horns, or the many horns, of the so-called antinomies. They are not antinomies really, but so-called antinomies, like, let's say, adaptation, yes or not, or chance, yes or not, or determination, yes or not, and so on. This is a very wrong way to approach living systems, as it was shown this morning also by the beautiful presentation of Professor Lloyd on orgasm. Is it adaptive or not adaptive? It's very difficult to make the choice. You should not make the choice because the two horns of the antinomy are generally contemporary and operate in that. Now, many people and many theories actually were channeled into two opposite positions, opposite general position. One example of this is what happened, for instance, it's a very funny case, in my opinion, what happened in the work of these three people who had the same Nobel Prize. Of course, one was of the head of the department, that was Wolf, and the other two were the ones who were working, make experiments to see whether they could find out the way to have a knowledge of a regulatory system of a living system, which was a bacteria in this case. They did find the first regulatory system ever found. They had Nobel Prize, both of them, actually three of them, and they took from this observation, this discovery, two very different ways of looking at life, at living systems. Two very different ways, particularly the two experimentalists, which were Monod and Francois Jacob, had two completely different visions. And this is what's said. For instance, Monod, he was saying that living systems are endowed with a project. He talked about teleonomy. He considered DNA the fundamental invariant of life. Now, fundamental invariant is a very heavy way of considering DNA, because fundamental invariant is another fundamental invariant is, of course, God. So DNA was a little bit too close. His definition of DNA was a little bit too close to God. He was very deterministic, but he had this subdivision between chance and necessity, very neat and so on. A very different situation was that, a very different opinion was that of Francois Jacob, who said in comparative anatomy a fragment is not anymore an isolated element. It is a sign of a whole organization. It has no program then. It's an organization. It's a dynamic organization. The relative relevance of an organ is measured through the constraints that it imposes to the others. So interactions between components of the systems. Everything is allowed as far as variation is concerned. Only combinations satisfying functional needs of life are allowed. Not directed by a program, but functional life. It is a play of interactions of organism and environment. It is the dialectic of the same and the different within a unitary history of nature. A completely different vision from that of Monod, completely different, of people who had been working on the same object and had the same Nobel Prize for that object. This is very interesting, at least to me. And, well, the opinion of Monod is very well represented in this thing which is the central dogma. You know that, well, I, with Gould and with many other people, don't like very much the word of the meaning of dogma in the case particularly of living systems, because dogma is a completely deterministic way of looking at life, and the informational metaphor which came out of this central dogma, this opinion, has been going on until now. Of course, it's in the daily papers, it's everywhere, and we always keep thinking about DNA as a program, completely deterministic program, and so on. Nothing bad in that, but it's wrong. Now, the basic concepts of the modern synthesis were absolutely coherent with the central dogma, in which they were either driven by chance, complete chance, and not the randomness that Longo was talking about, but complete chance, but the phenotypes were completely determined by the DNA, by the genes. So additivity was what the term used for the organization of living things, which were not considered connected one to another, but independent from one from another, as they are in a car, for instance, in any machine. So evolution was evolution of the genes, and not of the phenotypes, not of the whole thing, and so on. You know all these things we have been talking about this morning also. And three fundamental processes: new evolutionary change, mutations, chance, random drift, chance, natural selection, necessity, complete chance, and complete necessity. Gould had a very different way of approaching and studying science, and he was very interested in the former work by Richard Goldschmidt, who had been a very well-known heretic for a long time, I would say together with Barbara McClintock, of which I will be talking in just a moment. And he said about Goldschmidt, he actually broke sharply with the synthetic theory, however, in arguing that new species arise abruptly by discontinuous variation or macromutation. He admitted that the vast majority of macromutations could only be viewed as a disastrous, these he called monsters, but Goldschmidt continued, every once in a while a macromutation might, by sheer good fortune, adapt an organism to a new mode of life, a hopeful monster in his terminology, which I think was the first way of describing a jump in evolution, by Goldschmidt, of course, but taken also by Gould. As a Darwinian, he said, I wish to defend Goldschmidt's postulate, the macroevolution is not simply microevolution, the things that we have been discussing the whole day today. He was a very good friend of Barbara McClintock. Actually, they had two talks at the Cold Spring Harbor in 1951, if I recall well. And they had two ideas, concurrent ideas. One was the idea of the hopeful monster by Goldschmidt, which meant interaction between elements, components of a living being. And on the other hand, in Barbara McClintock's work, it was the discovery that the DNA was not stable. The DNA could change a lot for the present of transposable elements, which she didn't call them transposable because she didn't have that name, but she said there were elements which were jumping from one part to another part of the genome, so could give fluidity to the genome. The genome was fluid, was moving, was changing. The two things went together. They were good friends. They helped each other because they were both heretic and not the other people didn't like them so much at that time. Now, Gould actually directly talked about complexity And he says also the mathematical analysis of complex systems composed of multiple independent paths shows that the small perturbation can produce profound effects because of the way it cascades through the nonlinear interaction of the system. If you then add a little bit of randomness, you get profound and unpredictable effects. Now, this is the, say, general definition of Gould's view, which was very similar to a definition by physicists of complexity. These are two physicists who were actually studying complexity after 61, the beginning of this kind of area of thought, and they said the same thing that actually Gould had said: minor disturbances lead to intermittent events of all sizes. These events organize a system into a complex state that cannot be reduced to a few degrees of freedom. This type of punctuated equilibrium dynamics, they were physicists, but they were using the same terms, has been observed in astrophysical, geophysical, and biological processes, as well as in human social activity. Now, this is a complete change of idea from Monod and so on. So, well, you had, I'll skip this because you actually saw it many times, you know very well, the punctuated equilibria idea conception says that there are stasis and then there are fast changes in certain moments. Now, what I would like to, and this you also have seen this morning by Eldredge, and what I would actually now show is to look at the processes, at the molecular processes which do induce punctuated equilibria, punctuated changes. For we know much more. For at the time of punctuated equilibria hypothesis in 1972, very little was known about genome changes, complete genome changes, general genome changes. But now we know lots of them. We know very much about them. So we can say now that there are several processes which can accelerate evolution. And accelerate evolution means exactly what Beppe Longo was also saying, that randomness is what is needed for having complexity and for changing the evolutionary trends. There are several different events which can accelerate evolution. I will show you some of them. Now, mutations in pleiotropic genes. Pleiotropic genes, I think you all know what are single genes which are connected with many others. Now, the change of one of the head, let's say of the hubs, that have many others around, will change the whole system. Now, second thing is horizontal transfer. That means insertion into an organism, a gene which has not co-evolved with the genotype, with the former genotype of that organism. Often, if natural horizontal transfer happens, then you have changes in the whole organization or in large part of the organization of the receiving object. Then interspecific hybridization, which is much more common in plants than in animals, but it involves complete reshuffling of the whole genome. If you have interspecific hybrid, I show it to you, you will have shuffling of the whole genome, huge changes of the whole genome. And response to external stress will induce methods to have more randomness. It's the answer to the by internal stress, I mean what happens, for instance, in horizontal transfer. You put a new gene which was not there. This gives an internal stress because the equilibrium of the whole system changes. And that the answer to these changes from outside, let's say, or from inside, is to have more variability. And there are molecular tools, both in bacteria and in higher organisms, to have more variability. And that more variability happens afterwards to adaptation, but not in a single line, adaptation in some way to the new internal or external conditions. The two things are actually true. And also what I call dragging mutations, which means that there are some mutations which may in their own have an adaptive value, but only if they are in a concerted situation. We, the humans, are the ones who are actually using this kind of jumps, this kind of process for jumping into a new thing. All these factors that you can't read here, but most of them are actually situations of stress. In all these situations, you have an increase in variability, and the increase in variability is the main point, the main tool to adjust to a new kind of organism with new randomness. Now let's start with pleiotropy. Pleiotropy is a very obvious agent of change. For if you change one of these nodes, for instance, this node, which is a central node, this is node is connected with the other modules. Our organization is a modular organization. Our living beings organization is a modular organization. So if this one changes, also the others have to find a new equilibrium, a new dynamic equilibrium, of course, not a new static equilibrium. So they will probably change. And this is the case, there are many cases. This is a very well-known case which Goldschmidt loved very much. These are homeotic mutants. If you homeotic genes are actually genes determining the shape and the development of animals, all animals, also our development is determined by the same genes that are acting in the case of Drosophila. If you have a mutation that can change the organization of the organs, of the disposition, and they will change the whole organism with a single change in a hub of this kind of organization. So this is one. This is another possible thing which has been studied by Lewis, which said that duplication and diversification of homeotic master regulators underlines the evolution of an increasingly complex body plan, which means that we have genes are not equal, but there are some genes which are more equal than the others, as somebody would have said, and those genes are more equal than the others because they are hubs. Or they're interacting with a very, very important point of the organization of the organ. Many of those have been shown by our friend Minelli many times in his beautiful representations. Now, how do bacteria and how do higher organisms respond to stress and increase variability? Two different systems. In the case of bacteria, you have a single gene, for instance, his name is RPOS, which will induce, in case of stress, other genes which will replicate DNA with wrong mutations. They say that they will replicate DNA with mistakes, which means that the frequency of mutation frequency, then the variability, genetic variability of the bacteria will increase in that case. And the bacterium hopes that within this diversity, this new diversity, there will be something which will help him to resist to the stress. This is general in bacteria, to activate mutator genes, they are called, and other genes which will change. They activate DNA polymerase 4, for instance, which will make the DNA, replication of DNA, but with many mistakes. So they increase the number of single mutations in single nucleotides or single groups of nucleotides in single genes. This is the way bacteria do that. With the higher, yes, then there is another, oh, I skipped this. There is another way which is similar to pleiotropy, which is horizontal transfer. As I said before, horizontal transfer means a gene which is introduced into animal or a plant, but it was not in the genome of that organism. These are many, many cases. The most known cases are well, in all organisms. As a matter of fact, we studied this in our department a long time ago, and we used say wild or natural genetic engineer, which is called Agrobacterium rhizogenes. Agrobacterium rhizogenes about 4 million years ago or something, integrated genes from its hormonal system into plants of the genus Nicotiana. Those genes interfered with the hormonal system of the plants, of the different species, changes the organization, change the phylogenetic of the whole genus, according to which genes of the Agrobacterium were incorporated steadily, or not, or which complements of the Agrobacterium genes were in the Nicotiana plants. So you have a phylogenesis, which is this, here where you see the dots. That means that there are genes coming from the Agrobacterium, but in different complements. That meant that there was a diversification of the areals which were covered by the different genuses according to their physiological changed complement. So the insertion of a single or 2 or 3 genes from an Agrobacterium changed the evolution, drastically the evolution of the whole genus, completely changed to always to regain a new equilibrium. These are sort of many pleiotropic genes coming into a genus naturally, without the influence of man. And what is interesting is that if we look, as we did, at the phylogenesis of the same Agrobacterium genes in the plants and of the same Agrobacterium genes in the present-day bacteria, we found out that the genes which were in the plant were changed according to the phylogenesis of the genus. The others, the same genes out of the plant, changed with a different phylogenesis because they were in a different genetic environment, which was a bacterial environment, while the other one was the genetic environment of the plant, which means that all this means that there is an interaction, very strong interaction between the different components, as Gould was saying, and that interaction changed the evolution of the same gene in 2 different genetic backgrounds. Always accelerate, all these things, all these processes are acceleration processes because the change is accelerating evolution in different ways, of course. Now, we actually did something similar with a genetic engineering experiment. We introduced into a Nicotiana species a rat glucocorticoid receptor gene. We thought that that was so far from the plant that wouldn't interfere with the plant hormonal and phenotypic situation. But the first plants were very, very ugly, if I would say. They're absolutely unhappy, the transformed plants. They had few seeds, they were carrying few seeds, they were in a very bad situation. Then in 2 or 3, 4 generations of selfing of these plants without changing anything else, they reorganized some way, And you had two different kinds of species. Also from the morphological point of view, if you see here say the normal plant, I mean the not engineered plant, which is this one, and the engineered plant, morphologically they are different, but they have a different membrane, they have a different, a totally different reaction to insults, to external insults. For instance, they resist to pathogens, they resist to heavy metals, they have lots of these things. They have a different physiology. The hormonal complement of the plant, which has nothing to do with the hormonal complement of the rat, of the original rat, has been changed a lot, and so on. So there has been well, we didn't think about this thing, it could happen, but we had inserted a very far away gene, a gene coming from a very far away organism into this plant, and this is a sort of hopeful, really hopeful monster, because they are fine now, they were very badly off before. Now, let's try to go. Now, in animals, animals and plants, in eukaryotes, in higher eukaryotes, the response to stress is different and is epigenetically regulated, in the sense that we have lots of transposons, as you have seen in a beautiful presentation by Gregory, and we have almost 45% of our genome is transposons. Now, those transposons are actually blocked normally by methylation. Methylation means well, methylation means methylation of both histones and DNA, and they are completely blocked. In case of stress, they are deblocked, they are demethylated, and they start jumping. And when they start jumping, they break around the genome. They cause whole rearrangements of the genome. I'll show you some examples. I'll go back to that. These are, for instance, some transposons. Now, this case, for instance, is two maize lines of the same species. For the way, just two cultivars, two cultivated lines of maize, and in all those lines and in the two lines, a piece of say long sequence of DNA was analyzed in the two varieties, in the two cultivars. Here are the only sequences which are common to the two lines of the same species. These are only present in this line, and these are only present in this line. This has happened through reshuffling of the maize genome. Genes were the same, more or less, but they changed. But their position was completely changed, and changing position in higher eukaryotes means changing expression, because non-coding sequences are regulatory, which means that going in one place or the other will change the expression. These are a lot of other situations which are similar. In plants particularly, plants, as you know, as you all know, are able to make hybrids, fertile hybrids, while in animals it's very rare, but in plants it's very, very often happens. But when they make hybrids by replicating the chromosome complement, because otherwise they would have difficulties in meiosis. But in those cases, they lose a lot of DNA of both, and they rearrange all the two genomes. So the new genome, coming from two different genomes originally, is very different from both. They all change and they adapt always one to another. This is very well known now. There are many, many cases of this thing. For instance, here I just put a table where you see how much of the DNA of the two different species which had joined together has been lost after hybridization, and that goes up to 92, for instance. 92% of one of the genome has been lost. Just a few genes have remained in a certain case. Or in other cases, it's actually called shuffling of the genome shuffling. Also in animals, it's quite similar to the situation, because you see in Drosophila, for instance, increased rate of transposition in the specific hybrid happens. Rate of transposition means that the rate of transposons, which are one kind of the transposons, jumps. Then loss of retroelements methylation, as I said before. They are demethylated and they will be jumping. In wallabies, in mouse, amplification means replicating only little sequence of the whole DNA and spreading around the copies, which will go again and induce mutations in some places. Chromosome elimination in rice fish, in Medoca, and in marsupials, centromere instability in interspecific hybrids, which means, of course, difficulties in meiosis and complications in crossing over and so on. Very fast. This is only 1 generation, 2 generations, not very much, of complete change of the genomes. And the last thing is what I call dragging genes, but you may change the name immediately. It just came to my mind. Dragging because it drags all the other genes in a new organization. It's humans. For we are actually different from the other primates for a, say, small number of genes, probably less than 200 genes out of our 20,000, which are not taken away, but they change very fast, which have been accelerating their change only in our lineage. One of these genes is very important, is encephalin, which enlarges our encephalus. And you know that we are humans because we have a different ratio between the encephalus and the rest of the brain, not because we have a large brain, because the whales have a much larger brain than ours, but encephalin has enlarged the encephalus. And we are the ones in the world which have a higher proportion of the encephalus versus the other parts of the brain. The second is the dolphin. But the poor dolphin doesn't have hands. Speaks a little bit, but not very much. We do speak because we have FOX2, FOX3, FOXP2, FOXP3, all the genes of the Broca area. These are also in this area of the accelerated change. And if you look, it has been looked at the expression of the accelerated genes. Where is the expression of the general accelerated genes? And it has been shown that the high majority of those genes are in the brain, are expressing in the brain. So we are humans because only few genes started running like hell. I don't know why. One hypothesis may be that first we had a large encephalus, then we had to differentiate the different parts of that encephalus. And most of these genes are actually differentiating neurons. Many of these genes have to do with pruning processes. Pruning processes, you all know, means that we are actually losing synapses after birth. Only some synapses are actually maintained, and are those synapses which have signals from other humans. Not just signals from outside, but from signals from other humans, because the receptors, our brain receptors, will accept only signals from, well, not only, of course, but will accept very much signals from humans. In the brain, they are the ones who say, well, this synapsis is okay, the other synapsis is useless because it doesn't go into a reception channel, into a series of connections with the environment or with the thought. have no idea how that is. But so we have many more neurons. We have differentiation of cortex areas. Then we had probably the spreading of mirror neural systems and the configuration of comprehensive neurocognitive nets in different proportions in the evolution of our species and probably of the other three fellow species until to a certain point probably of evolution, which are, as you know, Neanderthalacensis, Floresiensis, and Denisovano. And okay, this is yes. These are yes. Another thing which is very important is that most of those genes which are expressed in the brain. There are very few genes, accelerating genes, which are expressed in the liver or in another place. But in those genes, most of the changes were in the non-coding sequences, not in the coding sequences. Also in the coding sequences, they were going faster, but more in the non-coding sequences. So our situation, and this has a reason. It's understandable. For we had the encephalus at the very beginning, like the other primates. What changed was the quantity, was the amount of encephalus in comparison with the rest of the brain. So it needed new non-coding systems, new regulatory situations of only these genes and not the others. Here there was probably a follow-up of adaptive situation once we had started our evolution, because this is true only in the human lineage. And Neanderthal also had similar situations, but he was not very. I think he didn't have FOXP2. I'm not an anthropologist, but I think he didn't have some of the speedy genes that we had in that case. So here are actually here are functions of some of these genes. Like FOXP2, of course, is very important for the language. And in fact, if you compare humans, very young humans, let's say one boy of 2 years and a half of age, with a chimpanzee of 2 months and a half or something like that, same age in terms of life duration, then they have the same characteristics. They both can count up to 3. They can distinguish 1 from 2 from 3. But the human baby has lots of more information from his fellow humans. This is why he changes very rapidly his pruning system. He organizes his brain in a very rapid way, but only if he can connect himself with other humans. It does not work when he connects with wolves who are actually breeding. In all those cases where one child has been taken by an animal, a wolf, any other, this boy will grow as a human being. He will have all the features of a human being, but he will be handicapped. For our receptors are specific to to human speech, to human words, to human faces. So all these processes that I've been talking about are all processes accelerating evolution. Accelerating evolution in the way Beppe Longo was saying, which means inducing variability, inducing new variability, abrupt variability in case of stress or not. It's faster in the case of stress, it's slower without stress, but it's always acceleration of a part of the gene, selective acceleration of genes. Probably their adaptive situation are important. So we can also say, as a lady whom I don't see here, but was at the former meeting on evolution in the genomic era. Well, she said that she was a neutralist. She is a neutralist. I think a daughter of one of the main neutralists of 1961, or the beginning of Kimura's thing. So, but she said a very interesting thing, that now the neutralist position is new in a certain sense, because she says transposons jump in case of stress, and they induce neutral mutations, she said. But transposition is regulated by epigenetics. It's regulated, as I said, by methylation or demethylation. So it's a random process in theory. It's a neutral process, not a random process in theory. But afterwards, there is, say, before there is a deterministic action, because is the reaction to stress. Then afterwards, there may be selection. I talked to her, and she actually agrees. So we can be a neutralist and at the same time selectionist, and at the same time also Gouldians. It's quite possible and quite useful, because you learn more if you have different ideas. Thank you.